This is another one of our monthly research webinars. Thank you so much for joining us today. We do have some program announcements that we're going to get started on first before we get to our speaker and get to the presentation. So we will start with Dr. Travis Kabali, and Travis, take it away. And yeah.
Oh, thank you, Rhonda. I'll be very brief there. And those of you that were on the call early might have heard our conversation with Dr. Branch, but the research program, we're migrating a significant amount of information over to our public-facing landing page. Rhonda will send you the link. We do, as you all are aware, there is 7 days a week, we do major searches using about 10 different search engines to make sure that we have all World Trade Center research publications going back to early October of 2001. There are 1,230 publications now that are have been migrated to the landing page. We also have impact scores. Those are updated in real time that are assigned to each of the publications. There are filters or topic areas that assist the search. We will be adding to those, you know, significantly in the next few months to help you locate specific items, you know, in this database. And it'll, of course, as I said, be updated. Right now, we're updating at least twice a month, and we'll probably increase from there. So again, this is available to certainly researchers, clinicians, and stakeholders, and we will keep you updated on progress that we're making. We're delighted to have Dr. Wold and his team present today. I want to welcome you all on behalf of all of the World Trade Center staff and researchers and stakeholders. And I will turn it over now to Melissa Runyon for the introductions. Thank you so much and welcome.
Good afternoon and thank you. Dr. Loren Wold is the Associate Dean for Research Operations and Compliance in the College of Medicine at The Ohio State University. There, he also serves important training and mentoring roles as professor in the colleges of medicine and nursing. He received his undergraduate degree in biology and psychology from Boston University and his Master of Science and PhD in physiology from the University of North Dakota School of Medicine and Health Sciences. His laboratory research has involved the development of molecular models and cellular technologies that explore the environmental triggers of neurodegenerative disease and cardiovascular and pulmonary dysfunction. The environmental stressors his laboratory is currently investigating include air pollution, e-cigarette vapor, and World Trade Center dust. Today, he is sharing his findings on the effects of World Trade Center dust exposure on cardiac and cognitive functions. This afternoon, please welcome Dr. Loren Wold.
Thank you so much, Melissa, for that very kind introduction. Thank you also to Rhonda for the numerous emails and helping get this organized, and Dr. Kabali for the invitation. I'm going to set up my screen share here, and I'm sure I'll do it wrong, so give me a second. Oh, see, I knew I'd do it wrong. Okay, so we're good. Awesome. So thank you so much for the introduction, and you know, Melissa gave a nice background on the area of research. So my lab, I've been at Ohio State for 16 years. We're affiliated with Ohio State, originally at the Children's Hospital here in town for 7, and then moved to main campus in 2013. And my primary area of interest has been studying the role of the environment or the exposome on cardiovascular triggers. And recently, in the last 5 years or so, integration of the neurocognitive network and how that is connected to the heart. And originally, we had been looking at particulate matter as an isolated event, gotten into e-cigarette exposures, and then in collaboration with Dr. Mitch Cohen, who I know many of you know on this call, we began looking at the effect of World Trade Center dust exposure on rats. And so that's the basis of the project today. And I'm sharing this presentation with Dr. Matt Gorr, who's an assistant professor at The Ohio State, who's worked, who's really led a lot of the project, at least the data we'll see today. And I've left some time for questions as well, because I want to make sure that we're getting those questions answered as they come up. And listed here, of course, is the grant mechanism, which this was a 1-year project that was probably about 4 years of experiments and data that we put into the 1 year. So we're trying to maximize the use of the World Trade Center dollars. So many of you may see your name here listed. There are lots of publications that have been published on the effect of World Trade Center dust exposure on a myriad of organ systems. So obviously cancer progression, respiratory disease, which is self-explanatory, prostate cancer, PTSD, cognitive impairment, and depression. And what's interesting is the exposure to World Trade Center dust has resultant issues that are quite similar to exposure to many other particles in the air, if you will. I know there's plenty of people on the call today that are interested in exposure of isolated particulates and particulates individually and their effects. And so my brain sort of thinks of this in a heterogeneous environment of exposure to a myriad of particles. And I think that's what we'll see with the data we look at today with the World Trade Center dust exposure. Is we're looking at it in the sense of a very isolated but extremely concentrated exposure to particles within the air. So to give you a general sense of how my lab approaches this, and so we're interested in what are the potential central nervous system, pulmonary, and cardiovascular responses to exposure to World Trade Center dust. So we know from previous work from many of you on the call, there are resultant pulmonary issues that are evident in the clinical space in the first responders who responded to the disaster, but also in models of animal exposure to World Trade Center dust. And we've now started to look at the cardiovascular response. We're going to show you some data upcoming, and I'll preface this by saying we have one more long-term time point that we're waiting to approach, which is 18 months in rats. But what we're seeing is quite similar to what we see in our air pollutant exposure models. And so what we're trying to do is tease out the mechanisms whereby exposure of something in the air can then have an effect on the heart at a much later time point. So, it makes sense if we expose the animals and then we looked at them, a couple days, a week, or a month later, but we're looking at them at a very long time after exposure. And then, as I mentioned before, this area of cognitive dysfunction or Alzheimer's disease, if you will, we've been very interested in how exposure to particulates could potentially have an effect on cognitive impairment and resultant neurodegenerative disease. We've been very interested in how you can use the heart or resultant heart function as a surrogate to monitor Alzheimer's disease progression. And so those are it's a sort of preface for what we're going to see with the data coming up. And so these are some striking pictures from one of Murray's excellent papers showing the dust that accumulated. And you can see in the top picture, it almost looks like snow that has accumulated in this alleyway between buildings. And in the bottom, you see this picture of this, so striking to me, It looks like, first time I saw it, it looked like a tea set found underwater in a shipwreck, if you will, after years of rusting. But what it's actually showing is a tea set that is completely covered by World Trade Center dust. And so we know that our first responders were exposed to crazy high levels of particulates from this dust during those couple days after the World Trade Center disaster. And here, from Liu's work in 2002, is a general characterization of 3 different areas where particles were collected and all the different metals. And Matt is not going to show you today, but we have also characterized the metals and particulates from the dust that we use. So what is this dust? This is just a picture to show you. This is dust that we get from Dr. Cohen at NYU, who literally, as many of you know, went out and collected this dust on the days following the disaster. And what we do is we actually, in our animal model, we re-expose in a methodology called insufflation, which Dr. Gorr will talk about. But we take these particles and we re-aerosolize them basically and expose the animals to this dust for 2 consecutive days, and then look at resultant physiological parameters as the months progress afterwards. And so in general, as I mentioned, we expose the rats to the dust. We wait and look at time course assessments to measure various physiological parameters. And I'm going to hand it over to Dr. Gorr, who's going to continue on and explain more of the methodology and get into some of the data.
Yeah, thank you, Loren. I'm very happy to be here. Yeah, so these rats that we exposed were, they're called SHR, or spontaneously hypertensive rats. So we thought that would best model, older individuals who years ago obviously were exposed to this dust are now encountering other cardiac issues as well. So these rats have high blood pressure, and after I think about 20 months, they go into heart failure. So we think they're kind of a good model for what we're looking at. So we gave these, as Dr. Wold said, we gave these rats 2 doses of World Trade Center dust via insufflation, which is done just by putting the rat on anesthesia for a small amount of time. And we have something we insert in their trachea, and it's puffed into their lungs in kind of mid-trachea level. So these animals get about 2 milligrams, which Dr. Cohen has helped us with the math to determine that this is similar to other animal models, via body weight and size of these animals. And then I'm going to be showing you data where we've collected at 3 months post-exposure, and then also at 12 months post-exposure. And as Dr. Wold mentioned, we have another cohort coming. It takes a very long time, obviously. So we're really looking forward to seeing what the longer time points of exposure as they start to coincide with the development of heart failure in these animals as well. But we're starting to see things, and that's kind of the message today. We're starting to see things, at least in the cardiac area, start to pop up that are different in the dust exposed animals. So we're hoping to explore that further. Next slide, please. So this is our sort of layout of our plan, is that every few months we do measurements that we can do, without sacrificing the animal. And then we have a terminal time points, which we've done so far at 3 months and 12 months. So we're looking to continue that. And I'll go through each one of these methods as we get to it. Next slide, please. So the first measurement we do is with this machine on the left, which is a cardiac ultrasound. So it's kind of nice that this company, Visual Sonics, they've created a machine that allows us to do ultrasound, and it's meant for animals. It's meant for small animals. And It used to be that we'd have to find an old one from some cardiac lab that wasn't being used anymore, but now we're able to actually take very good measurements using this ultrasound, and they're starting to sort of rival some of the older studies looking at cardiac MRI. So this technology has really helped us, especially with some data looking at strain and that sort of thing of the heart. So the data up here at the moment are a 3-month follow-up. And so these rats were exposed to those 2 hits of dust 3 months ago. And overall, you can see there's not really anything we can see with systolic functional changes. That is the ability of the heart to its contractile ability. The heart rate is the same. The size of these hearts, which there's various diameters as well as the size of the chamber, which are those 4 on the right. There's no changes in their size, so they're not experiencing any overt hypertrophy. And their ejection fraction is also not changed in fractional shortening, which is related to that. That's sort of how much blood can, with each beat, can the rats pump. So next slide is looking at the systolic function at 12 months. So this is following each dot represents 1 rat. As we continue to 12 months, we're seeing that these values themselves are changed, but the dust rats are changing likewise. So for example, they have larger diameter hearts, and that's just part of a rat being much older and starting to get hypertrophic. But we don't see any changes with dust, at least with systolic function. But looking further, if you want to do the next slide, please.
Matt, let me actually jump in here. So
Yeah.
what you'll see are both the functional assessment, which are on the bottom, ejection fraction and fractional shortening, which Matt mentioned is the percent of blood that's pumped each cycle, but also structure. So we're interested in not only how does the heart change functionally and its ability to pump blood, but also how is it structurally affected. And one thing I want to note is, this use of the SHR rat, many people have said, why are you using the SHR rat? And the short answer is the reviewers requested it. Originally, it was using a general sort of regular rat, a spread dolly or whatnot. But they had wanted a more representative, they felt, model of rats here. And so we use that. I will tell you, and we're not going to show you data today on that, but we have actually begun a cohort of looking at the general spread dollies, or the control, if you will, rats. Because if you look at the basic first responder characteristics, the majority of them were most likely quite healthy at the time prior to exposure. And so we've been able to answer another question, which is, does the pre-existing health of the first responder or the rat in this instance potentially play a role? And so one thing that I will say is you do see some variability here. Each dot on these graphs is an individual rat. And so what we're able to do with colleagues who look mathematically, using mathematical modeling, we can actually look at the change in each animal over time at each assessment. And what we're seeing is that we are seeing a trend for potential changes that we're hopeful that we may really dissect out at the 18th month time point, but we're not quite there yet. I think we have a little over 2 months left on that last cohort. So I just wanted to jump in on that. Sorry, Dr. Gorr.
Yeah, thank you. So just take a break from function. We've begun to process the genes at the 12-month cohort, and we're finding, and this is a 3-month cohort, that 3 months is actually quite a long time for a rat. So even despite only being exposed, those 2 times, we're seeing lasting changes cardiac genes that are regulating reactive oxygen species. So the changes that you're seeing here, shown like a reduction in glutathione peroxidase, are and there's a trending, not statistically significant increase in NADPH oxidase 4. That these are showing that there's actually a problem with the balance of reactive oxygen species in the cardiac tissue, which is very important, and it's been linked a lot with things like myocardial infarction to have issues going on. So despite both these animals probably going, another 2 years from this time into heart failure, that the deaths seem to be quite a bit ahead at only 3 months after exposure. So shown on the right are, we looked at, these are qPCR, so these are gene level changes in a few things that really are showing the same story, that there's a misbalance of the oxygen producing and creation of harmful reactive oxygen species in these animals at 3 months. So we're looking forward to processing that at 1 year, which I was hoping to have for today, but we're still working on that. So we also are able to, and this shows both 3-month and 12-month, look at diastolic function. And this is important because this generally is understudied, but the thought is that we see a changes in diastolic function prior to systolic dysfunction or heart failure. So we found that at 3 months that they had an increase. You can see in the top middle there of E over E prime. So what does that mean? So E is a measure of the filling of the ventricle, and E prime is actually, I call it the wiggle of the heart. So as the heart gets stiffer, it doesn't move as much when we look at it via ultrasound. So this is showing that there's a stiffening of the heart occurring. Interestingly, at 12 months, and this we're again, a lot of this is still coming in, but we're kind of seeing a reversal of that. So we're interested to see what's going on, further with that. That there's not a very clear diastolic dysfunction, but it's definitely showing that it's trending in that direction. We would start to see that with the IVRT on the left too, which is the length of time it's taking to relax, but we hope to see that in further cohorts. So the next slide is showing the pressure volume loops. And this is what's great about working with animals, is at a terminal time point, we're able to actually insert a catheter that's measuring the pressure and volume within the chamber of the left heart. And we also look at the right heart as well. This allows us to study the tissue under our controlled conditions to be able to see what's going on, in the heart in a lot more intrusive manner, but it gives us a lot more data. And what we found from this is if you look in the top 4 there, the cardiac output is not changed. So these animals aren't in any sort of heart failure at 12 months post-exposure. Neither is the pressure they're both about 100 shown there. The ejection fraction, again, that's the sort of percentage of blood that ejected with each heartbeat. That's not changed comparing the control and dust. Excuse me. But what we found was interesting is these bottom two. So EES is a measure of what's called the contractility of the heart. And this is done by, if you see those loops on the left, that's the pressure. Sorry, you probably can't read that, but the pressure versus the volume on the axes. This is done by actually constricting the flow into the heart, which allows us to see a change in the afterload. And it gives us a measure of what is the pure capacity of the heart to eject blood. And we found that in these rats, there's a clear reduction in contractility. This is the same with the stroke work, or the preload recruitable stroke work, shown on the right. So what we're finding here is that at 12 months after exposure, the animals that were exposed to dust were they have reduced contractility, which is indicative of these animals going into failure at some point.
So I just want to interject quick here. I put in the chat that a rat normally lives around 2 years. The SHR rat, 2 years is about the maximum that they'll live. And each month in a rat is equivalent to 2.5 years in a human. And so what you'll see here is at 12 months, post World Trade Center dust exposure, we're right around the time frame of where we are now, being about 21 years out from the exposure of first responders. So I just wanted to throw that out there, that this is a little bit past that, but we are seeing the trend, the very significant, I don't want to say significant trends, but we are seeing that there are changes in contractility, not quite at 0.05, which we anticipate at 18 months will come unglued, but just wanted to throw that out there.
Yeah, and so we can move the discussion. We're starting to look, and this is really exciting to see the metals that are retained in these animals after exposure. So the thought is that normally the lung is able to get rid of metals and other things it's exposed to, but what we found in doing some analysis is that several of the metals that are sort of indicators, or that are found in the World Trade Center dust, are retained in the lungs of these rats. So again, each dot is a sample from a rat, from one rat, and you can see that, especially with titanium, it's sort of night and day that there's a retention going on here, and that's even with the log2 axis. So we can see there's a lot of titanium being retained. So we're working with Dr. Cohen to be able to calculate how we have measurements of the amount of dust we've given them, where we know how the concentration of metals within the dust, and we know sort of the weight of the lung. So we're able to determine how much of the dust is being retained over time. So we're really excited. We just sent the samples for 12 months, I think a few days ago. So we're really interested to see if even after a year after exposure, if these metals are still being retained. So we also, within the same animals, are able to look at pulmonary function, and this is done through an instrument shown with our student David, called the Flex event, which is in the bottom left. And what it does is basically measure the pulmonary volume changes, and we can look at that in response to a challenge with methacholine. And what that allows us to do is see if these rats are more prone to having asthma as we constrict their airway. And that data is shown in the next slide. So we found that it's sort of starting to see a picture where at an early time point, we're seeing a change, a reduction in the sort of the flexibility of these lungs, whereas at a year later, that's starting to diminish. And we're wondering, and we have other time, we're wanting to incorporate other time points here to see how the lung function is changing, with time. And the details for 3 months in the next slide will show that, there's a lot of data that we can collect with this instrument, and we're finding that there are definite trends in the changes. As I mentioned, so the resistance is altered, and this here is without a methacholine challenge, as well as some other factors that are really demonstrating that there's a sort of a mild asthmatic phenotype in these rats at 3 months. But if you follow on the next slide, I think is the 12-month data. Now, interesting, I added more here because we received a sort of add-on to our machine that allowed us to take even more measurements. But I think it's clear from that there's no sort of overt lung dysfunction in these animals at this time point. So despite there being some retention of metals, at least for 3 months, that we know, that we're interested to see how this continues to play out. And that goes to something that Dr. Wold mentioned a lot of sort of particle toxicology experiments here. you're giving them a particle and examining them, the next day. Whereas we're interested to see what is the long-term impact of these. And as Dr. Wold mentioned, 12 months is quite a long time for a rat. So anything to add there, Loren?
I think what's really important here too, and what we've looked at in other particulate matter exposure studies, is the potential indirect and direct effects. And so, what's really intriguing, I think, with this pulmonary lung data, even though we're not seeing overt functional changes, is that we're still seeing those resultant contractility heart changes. So, we're always interested in how is the lung responding to exposure to a particulate, and this sort of stands, or at least points in the direction that it's the lungs are fine, even a year out from exposure. So we're trying to tease out the direct and indirect effects. What I mean by indirect effects on the heart are the particles and their exposure. Do they affect the lungs and cause the lungs to release some sort of response element, oxidative stress markers, inflammatory markers, etc. And so, it looks like the lungs, though, are at least normal here. The control and dust don't seem to be very different.
Yeah, and then in the next slide, we are seeing at 3 months a slight bit of pathology, and this is in the lung tissue. The alveolar density is decreased, and it's confusing, but that's this thought of mean linear intercept, or MLI, which is sort of a measure of alveolar density. But I think just looking at the images on the left really show kind of what we're seeing is there's been some damage to the tissue. So we're excited to see if this is still the same at 12 months and at 18 months as well. And then apparently, so we're working with someone who's looking at the behavior of these animals, and this is looking at anxiety behavior shown here, and each dot is a rat, and you can see we've learned a lot about behavior, that it's the standard error is usually quite large. So we're adding to the numbers of this to see if there really are true differences. And we're also hoping to correlate that with the amount of dust that they're exposed. So because we know we're able to calculate that number, we can look to see if that, if they had a little more dust, if their behavior is changed for that. But what this shows is, at least at 9 months, so we have the 12-month data that we've got recently as well, but is that there's no changes in these anxiety behaviors that are sort of standard protocol for rat behavior. We've also learned that it takes a very long time to analyze these data, because you're really sort of videoing the rat and timing sort of everything that it's doing within that time.
So we'll also be looking at a whole host of other behavioral tests once we do finally do the final time point, and also looking at potential protein markers of neurodegeneration within the brains as we process these animals. And so, that's the problem with really long-term studies in these animals, is you have to account for the aging process in addition to any potential exposures. And so it's been a host of hurry up and wait studies that we have planned that will be coming also once we take these animals, or we sack these animals. So, where are we with, we've shown you a lot of data. A lot of it is suggesting the potential detrimental effects of World Trade Center dust exposure. But where are we going from here, really, and what are the remaining research questions? So we know that, we know first responders are suffering from a myriad of age-related diseases following World Trade Center dust exposure. But one question, as I alluded to earlier, is how will these diseases be exacerbated by the long-term effects of World Trade Center dust? Another question we're interested in is the unique components of the dust and their potential to cause long-term effects to the heart and lungs. And then, of course, that begs the question of how does this potentially change the strategies for treatment of those exposed? We know that the 9/11 disaster was an unmitigated environmental disasters. So what potential measures, at least when thinking about the heart, lungs, and brain, can be put in place to prevent detrimental effects from those exposures? And so, I think of the 9/11 exposure as this is a very unique opportunity. When thinking about this, obviously it's difficult to find opportunities in disasters, but it's an opportunity to look at a very concentrated time, short window of time, and its potential to have lasting effects. And so, one thing that the lab has really been interested in, of course, is the connection between the heart and brain. And I reflect back to my collaborator, Federica Del Monte at Medical University of South Carolina. And what Federica has shown is that you can actually see, beta amyloid, which is the protein that is found in the brains of patients diagnosed with Alzheimer's on autopsy. You can actually see that beta amyloid in the heart. And so what we're interested in is how does the heart and brain potentially, how do they communicate? And there's some literature suggesting that there's beta amyloid protein that resides in various organ systems, and it's there as a normal process of aging. But how is that potentially affected with the exposure of World Trade Center dust? And so we'll be able to see that at a later time point as well. And then that begs a question of, are there early signs of potentially altered cognition or the Alzheimer's phenotype, if you will, post World Trade Center dust exposure? And so what we're interested with our air pollutant exposure studies is how can you develop or design a study to monitor changes in the heart and predict whether an animal or human will potentially develop cognitive changes? I reflect back personally with some folks in my family who developed what later became Alzheimer's disease. And both of them actually developed cardiac issues several months before we noticed any sort of neurocognitive changes. And so that's really the lens of which we're approaching this, is how can you interpret and use heart function as a predictor of potential changes in the brain? Matt, do you have anything to add there?
Yeah, I think we've been looking at these particle toxicology for more than a decade now, and Dr. Wold and I, and it's very interesting to see how different this dust is from other exposures. We're comparing this a lot with the particulate matter that one would encounter just with air pollution from motor vehicle traffic, and as well as we're working with particles that you get just from vaping. And it's the thought that there is retention in these lungs for a long time. It sort of really concerns us as to what's going to develop in the future. So we're really passionate about trying to figure out exactly what's going on at this molecular level for us to be able to sort of predict things for the future.
So I just want to thank the folks who actually do the everyday work within the lab, really led on a day-to-day basis by Dr. Gorr, who has been with me for a very long time. Dr. Gorr was my first PhD student who then left to a fancy postdoc in California, and we were able to recruit back as faculty. So really leads the day-to-day operations and studies of the students. Collaborators, of course, Mitch Cohen, I'm not sure is on today, but has been collaborating with us on this for a while. Dr. Falvo at the VA, who's the co-director of the burn pit exposure group there, and Dr. Combs, who's our Alzheimer's expert and former professor of mine from graduate school. So it's kind of fun to work with the folks that really trained me in the past. And then our funders, past and present, who allow us to do this work. And some fun pictures, because you have to have a fun picture. And so we try to get together for one nice event per year and celebrate the successes as well as the failures. And it's always fun to celebrate that over a nice steak. So with that, I'm happy to answer any questions you may have.
I'd like to thank you both, Dr. Wold, Dr. Gorr. Thank you so much for your sharing your findings, and then also the questions that you're continuing to find answers for. So it looks like we have our first hand up. So.
Dr. Reitman. I'm not hearing you if you're talking.
Dr. Ryman? Oh,
Sure.
oh, well, we
Yeah,
see
I know.
you now.
Exactly.
We see you.
Hard at work in the lab. That's great. Dr. Thompson, do you want to go while we're waiting for Dr. Reitman?
Yes, that was a really, really interesting presentation. I just had a couple of questions. First, how old are your rats when you expose them, and how does that relate to the human exposure?
Yep. Matt, do you want to respond?
Yeah, they're 3 months old. So there's been a lot of discussion about trying to relate an age of a rat or a mouse to humans. So at best, it's sort of a speculation that this is probably a middle-aged animal, and probably 30 to 40, maybe. That's a little younger than middle-aged. So how does that relate? Yeah, I'm not sure. Dr. Wold, if you know the age, I'm sure someone else knows the age distribution.
Yeah, so it's interesting because it's easier to predict the age in human years of a rat as they get older. So at 3 months, they're around 27 to 35 when it comes to human years. The problem is that these are SHR rats, and so they develop hypertension around 30 years of age.
Okay. The second question was your exposure route. You said IT, and then you said puff of air. I'm familiar with IT in a vehicle like PBS. Could you just elaborate a little bit more on that?
Yeah, there was not a vehicle used in this. It's insufflated, so there's just sort of a puff of air with a syringe. Okay.
And did you do any kind of clearance study with the lung? I thought it was really interesting. The pathology was quite striking. The damage is this retained in the lung, the dust, or is some of it cleared, or did you have any data on that?
No, we don't, and I think that's a fantastic idea that we could do with this model. Yeah,
and we do know, as we showed, that the metals are resident after 12 months, but we haven't actually. I believe Dr. Cohen has done some studies in collaboration with Won-Chi Chen at NYU, where they've shown some resident dust infiltrates, but I'm not exactly sure on if those exact clearance studies have been done. So great suggestion.
All right, thank you very much.
Thanks. Dr. Reitman, are you ready?
I'm fine. Sorry, I was on double mute on a different system. Sorry. Thank you very much for a very, very interesting talk. And I think that there's a lot of information that you're going to be deriving from here that'll be really informative. So thank you for that. I just have 4 things I want to say. first is a comment, which is that and of course it's going to come from me, but you show a beautiful picture of somebody's home. You show a beautiful picture actually of a coffee set that's completely covered by dust. And so I would just like to reinforce that when you discuss this, that you're discussing exposure that is both to responders as well as to people who lived or worked in the area. So, you know, just for clarification there. But it also has very interesting implications for something else you said, which is that, you know, one of the important points you made is that you're looking at an assessment of presumably normal animals. And and it pertains to the earlier question that was raised, which is, you know, what about those who are more vulnerable? And will you begin to see cardiac changes or pulmonary changes in the more vulnerable, for example, the older population or those who have other issues? So I think that would be very interesting. Finally, actually not finally, a third comment, really interesting about the metals that you find in the lung. And in fact, we actually published in human studies, Kaplan Shaw is the first author on that, and we did those with lung chi as well, that many of those same metals were still in the lung in the humans a long time after. So I think it's really interesting. Plus, we also showed some of alveolar destruction like you have. So I think that's really interesting to be seeing it in the rats. And finally, I just don't know, are these rats actually a good model for asthma? Do they get asthma? Or is this, I don't know enough about these rats, so I just wonder if you can comment. Thank you.
That's a great, first of all, thank you for your work. We've cited you and we appreciate all you've done. And that's a great question about asthma. So we actually have a mouse asthma model that Matt has helped develop within the lab, and it's an exposure of various bacteria over a time course of several weeks. I know that you can do this in a rat, but we have not done that. And I don't know of any rat models that develop a spontaneous asthmatic phenotype. Matt, I don't know if you have any.
Yeah, I think as far as inducing asthma in these rats, these rats were developed for the cardiovascular phenotype. So I'm not actually sure if there's much data on the asthmatic potential. I think the rats that they would use in asthma models, that they're on this background, I believe. So I think that could be possible. Yeah.
Great, thanks. Dr. Brackbill. Dr. Brackbill, okay.
Oh, there we are.
There you go.
Okay. Yeah, so I'm sorry if I had missed this, but I was just kind of curious about the controls, control group. And actually one question I kind of had is, so you used WTC dust, so-called WTC dust. Did you try sort of a non-WTC dust? You know, I mean,
Yeah, great question.
Yeah. I mean, assuming that dust in itself does the problem, or was something special about the WTC dust?
Yeah, no, awesome questions. What I'll say is the control for the SHR is called a WKY, or a Wistar coyote rat. So it's it has the same genetic changes, but it's a frame shift. And so there's actually, I would argue, as a cardiovascular physiologist that's worked in rats for a long time, that they're not necessarily the best controls, hence why we're doing this in spread dollies as well. But we don't have those results yet. And awesome suggestion about the basic dust. We have not done that, but that's a fantastic suggestion to do just a regular sort of commercial dust.
Okay, yeah, thank you.
Thank you, great question.
Yeah.
I think Dr. Ridley was next.
Not a doctor yet, but thank you very much. I'm just
I
curious.
promoted you for the day, so.
No, I was just curious because a lot of what you're talking about in response to all of this, like heart and lung examinations, was trying to use it as possible precursors for neurodegeneration and or cognitive impairments. So I was just curious, did you do any like beyond the behavioral assessments at, I believe it was 12 months or something, did you do anything like check cortical matter for cortical neurogenic atrophy of any sort, or?
Great questions, great suggestions. What we've done, we're waiting until we take the 18-month cohort down. And then my collaborator, Colin Combs, who's the Alzheimer's expert, neuro expert, we will assess all 3 of the time points at the same time. So we'll be doing all those studies. We're just kind of in a holding pattern until we get to that 18-month time point.
Cool. Thank you very much.
Thank you. Dr. Sundaram.
Thanks so much. That was a great talk. I was just curious. I mean, I may have missed this, but how was the installation of the dust performed in the rats? The reason I ask is, what was it with normal saline or PBS that you dissolve the dust in that changes the physiochemical properties of the dust and can affect your result?
Yeah, that was our concern as well. So we actually did something, just insulation is what it's called. It's kind of an uncommon word. So it's forgive me struggling to pronounce it, but yeah, so we have a measured amount of dust, and then it's actually just puffed into the trachea. So they're sort of intubated with a small piece of plastic, and so we're able to. We worked actually probably too long on trying to figure that out, but we're able to sort of show the amount delivered with each just by weighing it before and after, and that's right.
And a follow-up on that. So when you do that, given the nature of the dust, because of the very large particulate matter, right, and some have smaller, I mean, we're doing similar studies in mice, but we are dissolving the dust.
Yeah.
Question is the deposition pattern within the lung? I mean, that would matter, right, in the airways versus in the alveoli. Have you done, microscopy to see where exactly it's being deposited?
Yeah, I think that's a great suggestion, and we have the samples from these. That's something that we could look into, and that's very important, I think, to determine that. And I think what's nice about using a rat is the larger dust particle sizes are less obtrusive. So because the trachea is much larger, so we're hoping that that's an advantage of this model. Yeah. But yeah, I think that's something that we could very easily look into is how deep into the lungs, so to speak, are we getting with this exposure.
And I do know that Dr. Cohen has done some of those studies. I'm just blanking on the time point that he assessed them at, but that's a great idea to do that, especially do the microscopy in these animals.
Yeah, it's interesting in the mouse models, when they dissolve the dust, the physiochemical properties change, it becomes more neutral. Although they've shown the acute exposures actually that just PBS is fine, and it does cause acute lung injury.
Yep.
Immediately. So
yeah, exactly. Great. Dr. Hall and then Dr. De La Hall.
Thanks. Really fascinating talk. I'll just mention that there are some epidemiologic studies that have shown associations of heart failure and COPD with cognitive impairment in the elderly. So this is to get at the mechanisms as you're doing with these animal models, I think is really important for this. And there's also a fair amount of evidence now that particulate matter air pollution is associated with cognitive impairment.
Yep.
Some of the hot things are to do better person-specific exposure monitoring. I'm actually doing a study at Einstein using these little portable monitors that measure particulate matter. A similarly hot thing is to identify the components of the air pollution, and there are now wristbands that are used. And so we might want to be able to be ready for the next disaster to be able to do person-specific monitoring.
Yep.
So my question was actually partly stolen by Dr. Brackbill about the components and the importance of identifying what are the components. Different types of particulate matter are very different from others. Our preliminary data in the Bronx indicates that most of the particulate matter exposure in New York City seems to be from burning diesel fuel, and that was not one of the sources that was the major source at the at the World Trade Center. And are you planning future research to try to, not necessarily World Trade Center dust, but with the particular identified components to see what theirs are, to try to say, well, this is actually a big problem and this isn't? Because that would be a big contribution in general if you were able to pull that off.
Yep. Yeah, great questions. I appreciate your comment about particulate matter and neurocognitive. We've been working in that space for a long time and have an R1 on that exact topic. And so that's really the lens we've approached this from. But your comments are extremely well taken and definitely things that we need to do. Great. Dr. De La Hoz.
Yeah, I'll try to be brief in the, because there may be some people on the queue. One, in these rats, I mean, you chose, I still don't have it very clear why you would choose a rat that is prone to developing, I mean, almost deterministically, not
Yep.
prone, that's not the word, is bound to develop hypertension as a model. But be that as it may, I might as well, I don't know, take advantage of it. The other thing is that if they do they do it by mechanism, I don't know, that involves the microenvironment with inflammatory markers and atherosclerosis, or like I'm trying to find a parallel in the human, and because there is increasing evidence and realization that the pro-inflammatory microenvironment of obesity and overweight, which is more prevalent in this population than in the general population of the U.S., and other cardiovascular risk factors, they also have an impact on airway, on respiratory health. So that, to the extent that you chose, as I said, a model that is bound to be hypertensive, is that a model that also has that particular microenvironment? And I think you mentioned something about future studies characterizing the inflammatory situation of these animals, but so I don't know if you have. And the other thing about the measurements of lung function, airway resistance, if you could expand a little bit more on that, what type of measurement that was, and do you have microscopic studies, for example, in distal bronchiolar cells and other things that have been examined in inhalation injuries?
Yep. Thank you for your excellent questions. I do have to say to everyone, I do have to get off in a minute to, I have a meeting with the vice president of universities, unfortunately. But I think Dr. Gorr can likely stay on. So my answer to your question of why the SHR is because the reviewers asked for it. And we were sort of told we had to use the model. So I do not think it's the best model. But it was in response to that comment. We do have studies planned in doing this with an obese phenotype, which I think is also really important with this cohort. And Matt can answer the question about lung assessment.
Yeah, we have normal rats that we've exposed just recently to this, so we're interested to see if there are any difference. And this has been a really important question and animal research, especially in terms of the strain of the animal. They're finding that when you do the same experiment with several different strains, that you can get different results. So what can we sort of iron out is the important differences here. We'd love to do some more microscopy studies with this, and that's kind of just slated for future right now.
I do have to jump off, unfortunately, everyone. Thank you so much. This was a pleasure. Your questions are outstanding. Thank you, Dr. Kapale. Thank you, Rhonda. Thank you, Melissa. And any questions, drop me an email. I'm happy to respond to any questions via email as well.
Thank you so much, Dr. Wold.
Thank you. Have a great day.
Thank you. There was one other question. Dr. Gorr, do you have just a few moments to answer the other question? I think the hand was.
Yeah, sure. Yeah, I'm happy to attempt.
Sure.
Yeah.
I'm trying to see who they took their hand down. Oh, Dr.
It was
Clausen, thank
probably
you.
me.
Yes.
Yeah, no problem.
Yes.
I have to go soon, so I wasn't sure if to ask, but yeah, we've done a bunch of studying of cognitive effects, and so what we've found is actually maybe that this isn't the best model for cognitive functioning. One of the struggles we've had is that a lot of the sort of cardiovascular, cardiac modeling hasn't really worked. We have a lot of cognitive impairment people who seem at least non-hypertensive. And when we've done a couple of studies of neuroinflammation, we found that the neuroinflammation is there. Sometimes it's associated with exposure, which is kind of what you're reporting, I guess. But it isn't super strongly associated with the cognitive impairment. So it's kind of wondering, I guess this follows up on on Jag's comment, maybe whether or not you're kind of presupposing your finding before you actually find anything. And maybe different models will give you different things.
Yeah, we're excited to see, how this other model this Sprague-Dawley rat would maybe have some changes there. So I think it's a tough question, because it's really difficult to be able to iron out the differences. And you go so far with one model and you think, oh, I wonder if this one's slightly different and what's going on here. But definitely having that genetic predisposition for the high blood pressure, it's hard to iron out what's adapting here, over time versus what's been encountered as an insult. And whereas if you give them high blood pressure, as they're adults, for example, with you're able to say, well, this is actually adapting solely to the high blood pressure. It's not an adaptation of an adaptation, so forth. So
yeah, are you aware of what the comment of that reviewer was? What did that reviewer have in mind when they thought that her rat had to be hypertensive? I mean, I would have thought, well, if the reviewer had said, the rat has to be obese, oh yeah, I can see that, knowing the patient population. But in a nutshell, do you have even any sense of what the rationale of the reviewer was?
No, I completely agree with you. I don't remember the exact rationale of the comment, but I think there is this thought of putting two things together. You have hypertension and you have a previous exposure, but I would have thought of that as being a second step after looking at sort of what's going on in a normal animal.
All right. Dr. Hall, did you have your hands up again?
Well, yeah, just a comment. It might be from the fact that the Sprint Mind study and a few other studies have indicated that uncontrolled hypertension is a risk factor for mild cognitive impairment, and they might have thought that they should push. He may have been on that and might have wanted to push it. It's certainly worth looking at, but I can't read the reviewer's mind or jump out, but that's a possible idea that the reviewer might have had.
Thank you, Dr. Hall. Didn't know if I don't see any other hands raised, but I don't know if there's any other questions for Dr. Gorr while we're still here.
If he had to, I will add something. Do these rats develop emphysema? In older adults, you observe, it's kind of a physiologic emphysema. I don't want to call it physiologic, but it's a little bit of a loss of elastic recoil and a slight trend or, increased MLI, etc. So how confident are you? Do rats develop the same? And so.
Yeah, I think at the time point that we show that it's a bit before they're developing really the severe issues that they will. In terms of emphysema, I don't think these animals would develop anything like that. In the pulmonary, we were just looking there with the dust exposure. But in terms of hypertension, we see really the overt sort of heart failure more at like 20 months for these rats.
And I believe Mr. Ridley is on again with his hand.
Yeah, so I was just running through the article a little bit while I was listening to some of the questions, and something I was curious about, because it was a topic that you had mentioned a little bit earlier, Dr. Gorr, that you had mathematically figured this out or had some people doing that. But I was curious about the nature of not just the amount of dust that you gave the rats, but the time exposure and why you settled on it as the way you did. because you guys were focused on the first 72 hours of inhalation of WTC dust, as stated in the article. And then you did 2 consecutive days of 2-hour periods of inhalation of dust, if I'm reading that correctly.
Yeah, so the paper is slightly different than from the data that I showed you. That has 2 hours of exposure. The amount of dust they're receiving in the data that I shared today is the same as that paper, but the time is much shorter. They're just sort of given a puff of that. But yeah, in terms of timing, we're trying to mimic really what's in the air during that time, especially for first responders. But as was pointed out, for really anyone in the area, we're going to have much higher levels before that dust is settled. So that was kind of our idea, our thought. And really, in looking at trying to mimic these sort of events, it's very difficult to be absolutely certain that that they're receiving that, how much dust each person is receiving and that sort of thing. And we're seeing corollaries to this sort of thing in like wildfire firefighters, for example, like they're going they're going close to it for a number of days and then coming back, and then we're trying to figure out how much are they exposed to, and that's very difficult. Yeah.
Cool. Thank you very much.
Well, thank you. I'm seeing a lot of thank yous in the chat box, Dr. Gorr, and I appreciate everyone joining us today. Wanted to give it one more opportunity if anyone had any other questions or comments before we round this out for the day. And seeing no hands raised and no one's come off mute. Dr. Gorr, thank you so much for continuing to stay on, even though Dr. Wold had to leave as well, and we appreciate all the information you shared. I do have a little announcement. Sorry about that. There it is. In the chat box, you will see that I just put the next webinar that's going to be coming up, and Dr. Federer is going to be presenting about internet-based psychotherapies for PhD symptoms in World Trade Center responders and survivors. And that's November the 10th at 2 p.m. It's a Thursday as well. So that invite will be coming to all of you that were invited to this one very, very soon. So we'll send that out. Another round of thanks to everybody who attended today. Thank you to Ann Riddle, who's on our team, who put some extra information in the chat box. And any other last questions or comments? Was someone trying to come off mute? Well, all right. Dr. Gorr, thank you very much again. We appreciate it. And thanks to everybody who attended. These things are becoming it's really important to share the research and share the information and have a chance to talk and ask questions and learn more and engage. So we are definitely grateful that you are all here today. So many thanks. Have a great day, everyone.